Okay, good afternoon, everybody. Good afternoon, of course, only if you are in Europe. Otherwise, if you are in America, good morning, I suppose. And for who is now in Far East, we can say enjoy your dinner. My name is Marco Monga. I'm in charge of Human Capital Department in IIT. And I'm very proud today to open this webinar, the third out of four we have organized thanks to the work of Silvia Mondinelli. Silvia is the boss of our alumni community, but she's not bossy, believe me. I will not be alone today and enacting the role of a host, and with me there is a very friend of mine who is Stefano Gustincis. He's a principal investigator here in IIT of our neuroscience team. And this series of webinars has the aim to analyze the impact of the COVID-19 pandemic on science from different points of view. And today we talk about the impact of the lockdown and the social isolation of the brain, on the brain. So now I leave the screen to Stefano, who will introduce us the special guest of the day, and see you later. Thank you.
So thanks, Marco, for the introduction. Let me share a slide. Here we are. Today we talk about the impact of a lockdown and of social isolation of the brain. Okay, let me quote our guest, that is Professor Danilo Bzdok. He says, never before have we experienced social isolation on such a massive scale as we have in response to coronavirus disease. However, we know that the social environment has a dramatic impact on our sense of life satisfaction and well-being. In times of distress, crisis, or disaster, human resilience depends on the richness and strength of social connections, as well as an active engagement in groups and communities. Over recent years, evidence emerging from various disciplines has made it abundantly clear perceived social isolation may be the most potent threat to survival and longevity. We highlight the benefits of social bonds, the choreography of bond creation and maintenance, as well as the neuro-cognitive basis of social isolation and its deep consequences for mental and physical health. We are very fortunate to have Professor Bzdok here with us today. He studied medicine at the McGill University, Université de Lausanne, and Harvard Medical School. He then began his journey within the world of brain imaging and system neuroscience in 2010. Danilo has received training in neuroanatomy and defended a doctoral thesis in cognitive neuroscience in 2012. Then he won the Grunenthal Prize from his faculty, the Frederick Wilhelm Prize, and the Borchers Medal from his university, as well as the Hans Heinemann Prize from the German Society of Psychiatry. Danilo then studied for a PhD in computer science or machine learning, and is principal investigator in the International Research Training Group, which investigates the neurobiology of pathological aggression and impulsivity. From 2019, he is associate professor of the Department of Biomedical Engineering at McGill Faculty of Medicine in Canada. So it's really a great pleasure having him here today. I just want to spend one word and have a gentle reminder to the audience that a question and answer space at the end of the talk will be dedicated to anyone interested in having more info from Danilo's work and the topic of the day. Please use the lateral field you can see on the screen to send your questions even during the talk, and we will be very happy to answer to those questions at the end of the talk. So please, Professor Bzdok, we are very happy to have you here and looking forward for your talk. Thank you.
Perfect. Thank you so much for the generous introduction. Let me share my screen. Okay. So everybody can see my main slide?
We see.
So my name is Danilo Bzdok, and I lead a team that has one foot in computational methods, and in particular machine learning, and one foot in systems neuroscience. And we are most interested in human-defining types of cognition. I work at McGill and Mila, and our mandate is to at McGill School of Medicine and state-of-the-art analytics as practiced at Mila Quebec AI Institute. And today, I have the pleasure to talk to you about some of our very recent research starting March this year on possible impact that social isolation has on the human brain. And quick disclosure, part of my research activity is funded by Google. What is the most defining feature for the human species? I think one serious contender for this admittedly big question is the complexity of our social systems. So the single property that distinguishes humans most from our closest ancestors, monkeys, and potentially any other species living on the planet is the intricacy and sophistication of how individuals interact with each other. That is the reason why we today probably have so large, computational powerful, but also metabolically it's expensive brains. There must be a specific reason why humans have developed such costly neurobiological infrastructure. And evolutionary psychology thinks that the increasingly complicated patterns of how humans have interacted with each other along the primate lineage is one important driver that may have led to a co-evolution between the complexity of the brain and the complexity of our social systems. So it is instructive to consider that in a certain point in primate evolution, individuals, so primates started to individually recognize members of their own species. So it's important to realize that if faces cannot be uniquely recognized, it's so difficult to acquire knowledge about a single specific individual. So this is things like personality traits. If we cannot unambiguously be sure that another individual is really the individual we were talking to days or weeks ago, it is possible to really accumulate evidence that allows us to infer others' personality traits, action repertoires, beliefs, and likely actions in the future. So this is one important ingredient that maybe was an early step in the increasing sophistication of social cognition in humans. And a second aspect, which a lot of scientists believe was key, is the capacity to introspect into other people's minds, sometimes called theory of mind, perspective taking, or mentalizing. So the ability to introspect into key for the emergence of language with a complex, it is also the basis of a number of capacities that humans have developed, such as writing knowledge in books, passing on knowledge from one generation to the next, building nations, interacting across nations, retributive effects of how humans try to improve in every generation, up to retributive effects where the cognitive evolution of the human species has surpassed the biological evolution by a great deal. So those are just some arguments why the complexity and the sophistication of social interplay may be a key property, or maybe the most human defining aspect that tells the human species apart from other animals living on this planet. So an important question that this begs is what happens when we take away this very important source of stimulation that that we believe has probably been key in the evolution of the human species, but also the expansion of the human brain and the higher association cortex in particular. So, and loneliness, perceived social isolation, is what happens when a human being thinks he or she does not receive the amount of everyday social interaction, interaction that they would like to have. So what's important to realize here is that we are not talking about the objective frequency or quantity of social interaction with others. It is really what a human being perceives to be sufficient or not sufficient. It is not just a simple basic emotion like happiness, disgust, or sadness. And loneliness is a complex psychological phenomenon that has both of a state or a trait. So there are momentary features. Lonely people may feel more lonely at a certain point in time, and lonely individuals have a systematically skewed perception of the world. Lonely individuals expect systematically to be excluded by others. Lonely individuals interpret social cues from others as more negative as they are. Loneliness has a sociological component. Lonely individuals are more likely to know other lonely individuals, and befriending somebody who feels lonely increases one's probability of also becoming lonely later in time. Lonely individuals tend to live at the periphery of social groups in our society. But the impact is not only on psychology. There are widespread consequences that we have studied a lot in medicine and psychology. Wound healing is slower, the immunological system is affected, and there are many other mechanisms and axes in the human body affected by this complex type of. What hasn't really been studied so much is the manifestation of loneliness in the human brain. So the difference is really that most research has been done either in animals or without studying the manifestations in the brain. So what we have done and what has been accepted for publication is the largest existing study on social perceived social isolation in the human brain. We have conducted the study in the so-called UK Biobank, which is the largest existing biomedical data set that we have so far. 40,000 of these subjects have high resolution brain scanning. So we know the volume of the various parts of the brain in 40,000 individuals from the general population. What we also know for each of these participants is what or no, do you often feel lonely? So we're asking for a chronic form of perceived social isolation. On this slide, you see on the border the various global network effects of structural differences in individuals who self-identify as lonely. So there is a number of large-scale brain networks that we know to dominate function and that form functionally cohesive groups across the entire brain. What is striking here to neuroscientists is that in B, the default mode network really sticks out in this Bayesian pattern learning analysis as the most relevant brain system that really explains the difference between who identifies as lonely and who doesn't. Why is this surprising? The default mode network is potentially the group of regions that have evolved most recently in history. They subserve the potentially most complicated forms of social interaction, as I mentioned, perspective taking and so forth. However, if you look at the pattern A, if you look at the region level of this analysis, what has really been emphasized in previous studies in animals is not the default mode network, but in the right hemisphere, components of the inferior parietal lobe and the operculum that you see in CO, parts of the stress axis. So previous research on loneliness in humans was very much focused on the idea that feeling socially disconnected is a chronic stress stimulus for humans. That explains why lonely individuals tend to have perturbed cortisol metabolism, which leads to poor sleep, which may in turn lead to poor cognitive performance, which may lead to worse memory performance. So there's various ramifications, but the overwhelming emphasis of trying to explain loneliness in the human brain has not been on the higher association cortex. However, if we look at 50 times bigger sample than has ever been studied in this area of neuroscience, we find a different conclusion than existing research, and that is that the default mode network, the highest integrative areas of the human brain, those really potentially explain most how a lonely individual is different from a non-lonely individual. So quick technical note, the modeling innovation here is that we really performed a pattern learning analysis in this population scale data set at 2 scales at the same time. On the right, we modeled the individual brain and designed a model that also models the overall influence of these groups of brain regions. And this is what you see here in panel B to H. And by simultaneously modeling the impact of separate isolated brain regions in brain structure, but also their grouped influence of how much the whole collection of brain regions impact loneliness. So groups of brain regions that we know to be functionally related to each other. It was by using this model that we really unlocked that the default mode network may be the most relevant source and the most important manifestation of loneliness in the human brain. So if you're not convinced, let me show you 2 additional windows into the brain that confirmed this intuition. So there are different modalities, different types of brain imaging that have also been performed in these 40,000 UK Biobank participants. This is functional brain imaging data that you see on this slide. So this measures the amount of functional coupling of different parts of the brain with the same atlas that you have seen on the previous slides. The overall networks that you see on the left on the y-axis are roughly ordered from the most advanced, from the highest integrative areas and the default mode network, as well as on the bottom. So going to the bottom where we have the least integrative, the most sensory, maybe most primitive brain areas. So what you see is that we have an increased functional coupling between the highest integrative areas, so the default mode network in particular, but also control networks and the emotional limbic system and their interaction with the default mode network. So one speculation is that what happens is that lonely individuals own to engage in mental imagination of social relationships and imagination and scenes from the past. So any means to imagine, to make up, to fill the social void by creating social events that are not actually happening in the immediate environment. Lonely individuals are known to maybe talk a little more to their pets, like their dog, called anthropomorphizing. Lonely individuals are known to have sometimes intense relationships with protagonists from sitcoms on TV. I could go on. So there's a lot of indicators that lonely individuals try to make up for the lack of social interaction in the actual reality by generating synthetic social experience from the inside. And that makes sense with this finding here, because the default mode network is known to be involved in mental scene construction, imagination, and reconstruction of experience from bits and snatches of memory. So these parts of the brain are furthest away from the sensory processing circuits that would process the actual environment. So this is one possible interpretation why we see this pattern. I also would like to direct your attention to the blue functional couplings here. So you see that the visual cortex that would process others' faces and gestures and mimic, it is actually functionally decoupled from the higher association cortex, including the default mode network. So not only is there a compounding functional modulation in the default mode network, but also sensory circuits appear to be functionally decoupled from cortex. So, and this could be related to another aspect that we know from psychological research in loneliness, and that is the skewed perception of social cues from others, as I have already mentioned. If you're still not convinced, here's a third modality, a third way to look into the brain in these 40,000 subjects. We looked at 48 major white matter tracts that are the most important in connecting the gray matter regions that we saw in the previous slides. And the three most important white matter tracts that really explain the differences in loneliness are all related to the same system, and that is the so-called fornix. You see it in red here on the top. The fornix is a very special white matter fiber tract because it really combines the axons that come from the hippocampus specifically, so the medial temporal lobe of the emotional brain, and these axons lead to the default mode network. So we know that these tracts shown here in red, they do have a specific relationship to the default mode network. And although we test a whole battery of different tracts, we specifically find that the top three are all related to the most relevant input source that feeds information into the default mode network that we already found in brain function and in brain structure before. Okay, so the conclusion plus intrinsic functional coupling, gray matter variability, and major white matter tracts, we find that systematically we identify pointers to the highest integrative areas and the default mode network in particular to be relevant in loneliness. So loneliness is only a part of social isolation. There's another aspect that is important in social isolation, and that is the objective amount of social interaction in humans. So here we are not studying anymore the subjective impression of loneliness, but how many people does somebody meet on a weekly basis on average? How many close friends do they have? How often do they share their experience with others? So this is what is captured by social support, which we also studied in the. So I would just summarize here. Again, there are 4,000 participants, and we see that it is not the higher association cortex, the default mode network again, which we see here in red. Instead, it's in purple, the ventral attentional system, which is sometimes called the saliency network. It is this system that is actually most relevant in explaining this objective form of social interaction. So this system is, for example, known in being directly tuned into the current situation, empathy, affective sharing, reading other people's emotion, gestures, and mimics. So it is more directly coupled with the action perception cycles that we would need if we really meet people directly in immediate encounters. So although we look at social isolation again, the aspect of social isolation is completely different here in this example. And we show that a different circuit is coming out, and that is the intermediate level saliency network, not the default mode network. So this is the same Bayesian hierarchical analysis. We look at separate levels at the same time. And here's the region level analysis, where we see positive in red and negative effects in blue of how social support differences come out in the human brain. So
Danilo,
we see several.
Danilo, sorry if I interrupt you. I think the background noise that we have is really difficult for everybody. What's happening? Do you have an alarm?
Yeah. Okay.
Oh,
yeah. Sorry, there's an alarm in the building.
Okay.
In the building.
Fantastic.
So
now it's better.
Sorry about that.
No problem.
I cannot control the alarm in the building.
Yeah, I know. No, clearly. Yeah,
okay.
Yeah.
Social support. So point is, social support is really the other side of the coin, right? So this is kind of the objective, the non-emotional aspect of social isolation. And you see that there's a completely different circuit. It's also pretty high in the neural processing hierarchy, but it's not in the most integrative the deepest layer of the neural network that we have in the brain. It is a completely different type of system. What we see in blue here is the orbitofrontal cortex as well as the anterior insula. So there are positive and negative effects that we see across brain structure that are relevant in social support. Okay. But several other aspects of diversity between humans really play an important role here. For example, the amount of everyday social stimulation is known, and we have shown, has a specific effect in males and females. So if you take something as simple as the household size, so that's just the question, how many people live where you live, in the apartment where you live? How many people are there? This could be friends, there could be partners, there could be children. And you see on the left that in the amygdala, the favorite brain region in psychology, that it is really in females that there's a big difference in the structure of the amygdala as a function of household size, but not so much in males, which is the box plots that we see here in blue. On the other hand side, the ventromedial prefrontal cortex, a different part of the limbic and the emotional brain that also came up here in social support. And there you see in blue that it's mostly in males that we see a structural difference of this measure of social stimulation in everyday life. So those are just two examples that, again, in the population scale data set, we can find quite strong and convincing evidence that there is a divergence of how the amount of everyday social stimulation impacts the human brain. It's also related to aspects of socioeconomic status. If you measure, for example, the amount of income somebody receives per year, you see that in males, there's really a big difference in the nucleus accumbens, which is the keynote of the reward circuitry. But we don't really see that in females. High income and low income females have essentially indistinguishable distribution of nucleus accumbens volume. A similar effect can be seen if you measure socioeconomic status as a function of healthcare. Does somebody receive public healthcare or private healthcare? So what you can see there is in males, there's again a big difference in whether or not the reward circuitry of the social brain shows structural differences or not. But we don't really see this in females. Why is this important? There is a connection to loneliness and social isolation, which is the following. We know that social interaction is intrinsically rewarding. So somebody who is less socially stimulated as he or she would like has a higher tendency of various forms of substance use or abuse, smoking, alcohol, other substances. So we know from animals that indeed, if there is a period of social deprivation, there is a tendency to substitute this intrinsic social reward that otherwise would be received from interacting with others, that this stimulation is maybe replaced by compensatory behavior and substance abuse. So, and just some last outlook slides. There is a pervasive difference of the social brain in males and females, which you can also see in this analysis here. So this is really important because we know that across life, females tend to have more robust, more solid circles of close friends. So males are more at risk to lose, especially their close friends and close interaction partners, the older they get. And this is why in the current period of social isolation, it may be the case that males are actually more susceptible to bouts and periods of social deprivation than females, because males, especially in older age or mid-age, older age, on average, they are less tightly integrated in social networks and support circles. So, and that this may be really relevant is what you see in this UK Biobank analysis here, where you see again that the most solid associations of aspects of individual differences and diversity, social, demographic, personality aspects, they map differently on the male and female brain. So it's easy to conclude that the current stress test in terms of social deprivation that we are going through in the COVID-19 pandemic, that this may differently affect the male and female brains. So here you see, this is the last data slide. Here you just see that you can map out, thanks to the population data set, how are these nodes of the emotional brain related to aspects of social behavior differently as a function of sex, but also age. So on the left, for example, you see that the ventromedial prefrontal cortex that we found in objective social isolation. If you measure whether somebody tends to worry about life, about social relationships, the structural manifestation of this varies as a function of sex, that's here in red versus blue, as well as age. And there is a pretty striking difference in how this brain behavior association plays out in these different strata of our society. So you can see similar effects in the frontal on the right, which is more related to the default mode network that we talked about at the beginning. So to summarize, loneliness is something different than social isolation. I try to emphasize that loneliness is really a complex phenomenon and a complex psychological adaptation that has both state and trait measures that impact the very moment, but also impact how people behave over time. And there's no other way than directly asking somebody whether or not they feel lonely. Social isolation is the other side of the coin, because we are asking how often does somebody really socially interact with others, and how close are these other individuals that they're interacting with. Second point, loneliness is really not just an emotional state. We emphasize very much that there is impact on face perception and various other aspects of the body, such as the immune system, sleep, cognitive performance, and even earlier onset of Alzheimer's. So there's the whole body is affected, not only the. We had an emphasis on stress, cortisol, and perceptive differences in loneliness in existing neuroscience research. But it's maybe only becoming clear now that the default mode network is maybe at the heart of how social deprivation manifests in the human brain. And the last point here is what's asked very often is what is the causal relationship of how loneliness comes about. Are individuals lonely first and then they get depressed and lose their friends, or is it the other way around? I would say that this is very hard to know given the data that we have from humans. There may not really be the perfect data set right now to answer this particular question about directionality. So with this, I would like to thank my collaborators on the left for the inspiration. I thank these institutions on the right for the financial support, and I thank you for your attention.
Thanks a lot.
Sorry for the.
Thanks a lot for your great and very interesting talk. And also, we are very grateful that you were able to deliver a talk in the middle of a fire alarm. So it was very, very self-confident from you. We really, you know, appreciate that. It was really, really very interesting. I'm just, you know, it's. I know that, for instance, Marco has some questions that I'm sure he would like to present, and then we can follow with more questions on the topic.
Yeah, waiting for the audience, I can ask my question. Only one point, Stefano. We didn't see your webcam on. Maybe you have some problem.
Oh, sorry. Okay.
No, no, okay. Don't mind. Now,
Great.
now I am on the stage, so you can go. No, I already heard something about my question during your talk, Danilo, and let me tell you that it was very interesting anyhow. And also one of your final statements told us that we have a lot of, let me say, research efforts to be carried out up to now to understand something more about the effect of this pandemic on our brains. But anyhow, I'd like to know your opinion and to understand if you think it's possible that long-term stressful conditions, such as a pandemic, can cause permanent neurological change in some people, especially when, and this is, let me say, my political add-on, especially when the policies of containment and management of emergence are less effective than in other countries. I hope it was clear.
Yeah, thanks for the very interesting question. I would say it is challenging to give a very clear and very specific answer. It is probably the case that, at least in recent history, we haven't undergone a mass social isolation of the scale and the amount of how acute this really is in all these various countries simultaneously. We haven't really undergone such a challenge in the recent history, probably. Even if we did, it is interesting to consider that we did not really have high resolution brain imaging or whole genome sequencing, let's say 40 years ago. And even if there had been another pandemic with country-wide social isolation measures, we would probably not have been able to measure it as carefully as we can now. So another important aspect I haven't mentioned yet is really sensors data, smartphones. So there's a much better way to study really this global experiment on social isolation that we are going through. I'm not aware of data in humans that have a very close relationship to what we are going through now. What I can say is that in monkeys, there have been causal experiments that did change the amount of social stimulation in the scale of weeks. And after just a few weeks, you can measure brain structural adaptations, plasticity effects in these monkeys. So as monkeys are our closest ancestors, it's likely that our brains have undergone adaptations due to the social isolation period too. And the brain regions that the scientists have found in monkeys are also related to what we call the default mode network in the human brain. So the monkey research pointed out very similar brain regions that are related to the amount of daily social stimulation that we also found in our loneliness research. And that's particularly important because the same brain circuitry is also believed to be the end pathway of Alzheimer's disease. So I think one immediate consequence and avenue for future research is that undergoing an acute social deprivation period, such as now, may actually increase the onset of Alzheimer's of thousands of people in the future. I hope this roughly answers your question.
Thank you.
I have a question from the audience. And what is the relationship between social isolation and social exclusion, if any?
Oh, yeah. How to say this? So okay, there's one specific thing I can say. So there is classical neuroscience research in humans by Naomi Eisenberger and colleagues in a classic paper 15-20 years back. And what they found, social exclusion, the experience of not being allowed access to being part of a social group, that affected the same circuit as what we found in objective social isolation. Okay, so we can say the circuit that has been reported for actually undergoing social exclusion, the same circuit has also is exactly what we found in our social support analysis. That was the second study I talked about. Yeah, that's just a different scale. So social exclusion can have many forms. That could be on the individual scale. It could be based on ethnicity. There's so many different types of social exclusion if you look at it from an anthropological perspective. The way we framed our research is mostly along this objective versus subjective social isolation. So the framing is not exactly the same, but we find circuits that have been reported before in social exclusion. That's what I can say.
And thanks for your answer. I'm wondering, what is the consequences? So you just told us about potential strategies for pharmacological intervention.
Mm-hmm. The consequences for medicine, for society, for therapy?
For medicine, for therapy, the consequences of treatment. Instance, yeah.
So what I can emphasize is what are aspects to avoid negative consequences, and that is a number of measures that we have pointed out in our paper with Rom Dunbar that I've cited several times. We know that there is a number of activities that prevent people from feeling socially isolated, and many of them can also be carried out in our current time, in our current climate. What's important is to realize that social networks in the digital world, they're very similar to the architecture of social networks in the real world. So somebody who, let's say, has mostly superficial friends, but a lot of them in the real world, will also show this pattern on Facebook. So what this means is that humans have evolved technological inventions that can carry regular social interaction in ways that were not possible before, maybe during the Spanish flu, for example. So, and what's very important is that there's a visual component. We know from previous research that it's not enough to call people on a regular basis, but it's particularly important to actually see other people's faces and emotion. As I try to emphasize, reading other people's faces is one of the most important skills that humans have evolved to do. And it is maybe one of the most efficient sources of environmental information for the average human being. So to protect information and stimulation through other people's faces, that is a very important component. And that is particularly important for the elderly, because our grandparents may be less accustomed, maybe less often use things like Zoom or video calls as we are doing now. So I think ensuring a sufficient amount of social interaction can be done in several ways. Technology really helps us here, but I think it's particularly important to think about this in terms of people in the third age, as we say.
Okay, thank you. Thanks for your answer. I don't see any other questions from the audience. Marco, do you have any other questions?
No, I think we can go to the end of this webinar. I thank you so much, Danilo, really. It was very interesting, and I learned a lot hearing your talk. Thank you so much, Stefano, and thank you so much, Silvia. And let me understand if I can share my slide, the last one. This is an experiment. Do you see?
Yes.
Hope so. Okay.
Yes.
Okay. Let me only remember to the audience that this is, as I told you previously, it was the third out of 4 webinars that we organized. The last one will be on December 9th at 4 p.m. Continental Europe time, of course. And the topic of the last one will be the political epistemology of pandemic management. And we have 3 special guests, 2 coming from Ca' Foscari University, Daniel Omodeo, and Charles Wolf. And the third one is Flavio D'Abramo, coming from Max Planck Institute in History of Science. And it will be the occasion to finalize this huge job and also to share with all of you the greetings for the next Christmas time. And we'll see what will happen in 2021. Hopefully something will be fixed about this current pandemic, and maybe the next webinars will be about the phase 2, the phase after the pandemic that we are living now. Thank you so much again, and see you next time, and have a nice evening. Bye-bye.
Thank you.
Thank you so much. Thanks for the invitation. Bye.